Hi there. This is the first part of a section on cancer, the evolutionary aspects of cancer, and you can find the material in Chapter 18 of the Integrated Molecular Evolution Second Edition. Cancer in general can be defined as a breakdown in gene expression control. So certain genes can change their expression either by increasing the expression or decreasing the expression depending on the particular gene, causing the cancer or the cell to go out of control. You might be wondering why I include cancer in an evolution course, and it's primarily because the mechanisms that cause cancer are the same ones that lead to many evolutionary changes. So this includes mutations, inheritance of those mutations, recombination events, some of which cause chromosomal translocations, gene duplications and amplifications, environmental exposures, and then mobile genetic elements such as viruses, transposons, plasmids, and introns. So gene expression in cancer, called oncogenesis, often involving oncogenes, is a set of diseases, not just a single disease, but most of them involve evolutionary processes that lead to these changes, including mutation, recombination, and amplification. And these lead to the cells failing to respond to their environment and being more or less immortal. That is, there's no apoptosis or cell death where there normally would be. So in other words, cancer is when there is an essential loss of gene expression control in those cells and groups of cells. So this is a simple diagram of some of those processes that are interrupted in oncogenesis, or the development of cancer. So the first is evading apoptosis, that is programmed cell death. So the cells become more or less immortal. The self-sufficiency in growth signals, so whether or not there's a hormone or other molecule telling the cell to grow, they grow anyway. And they're also insensitive to any anti-growth signal. So if there's contact inhibition, usually from one cell to the other, and that's lost in cancer cells. When they become even more dangerous is when they start invading other tissues and eventually metastasize, colonizing other parts of the organism. They also have limitless replicative potential. That is, they continue to replicate their DNA and divide, and then sustained angiogenesis. Many cancers are able to attract blood vessels to themselves so that they have a continuous nutrient supply. Here's a list of some of the characteristics of a cancer cell. First, they exhibit uncontrolled growth, continue to grow even when they're not normally supposed to grow in that position of the organism. Two is mutations of signal transduction pathways. Those are internal pathways which take a signal from the outside of the cell, move it to the inside of the cell, and then start affecting different pathways within the cell, including within the nucleus. Three is no apoptosis or cell death, so they're essentially immortal. There's a loss of contact inhibition, so even if they touch other cells, they continue to grow. Loss of anchoring to other cells, so eventually they lose those anchors and start moving around locally and then later more broadly. And then they have a reduced need for growth factors. They continue to grow when they normally would not grow. They become even more dangerous when they start to invade other tissues, so they'll then start to affect the functioning of those other tissues. The second is angiogenesis, or the ability to attract blood vessels to themselves. And then the third is metastasis, that is to start to colonize other parts of that organism and then affect those organs. And that's when they become very dangerous. As I said, there are many different types of cancers, and normally they're named by what sort of cell types they originate from. So carcinomas are cancers of epithelial cells which come from the ectoderm in the embryo. And these are cancers of skin, lungs, breast, prostate, pancreas, and colon, and others. Sarcomas are cancers originating from mesoderm and mesenchyme, or connective tissues, and these are primarily cancers of bone, cartilage, fat, and nerves. Lymphomas and leukemias originate from blood-forming cells, often arising from mesoderm and mesenchyme. These are macrophages, etc. Germ cell cancers are primarily from endoderm, and these are testicular and ovarian. And then blastomas are formed from immature cells, usually found in children, and can originate from ectoderm, mesoderm, or endoderm. So remember, in some of the last presentations, I showed this where it's development of animal tissue. And in particular, by the gastrula stage, there is ectoderm, which are the outer surfaces, endoderm, the inner cell layers, and then mesoderm, which originates from interactions between the ectoderm and the endoderm. And so cancer cells normally revert to their embryonic type interactions, and that can include rapid growth as well as movement from one place to another in the organism. Remember from last time also, or one of the previous presentations, when you have the mesenchyme in a blastula, some of these cells start to move around in early gastrulation. And by mid-gastrula stages, they not only are moving around in the organism, but they're acting on different tissues to cause them to grow into certain formations within that organism. So some of these cells, actually, when they become cancerous, do the same thing. They start to move around in the organism. That is, they metastasize easily. And they also have influences on other cells around them, such as rapid growth of the cells around them, as well as rapid growth of those cells themselves. So again, in the gastrula, you have these primarily three different cell types: ectoderm, mesoderm, and endoderm, and then the germ cells also, which are set aside, as I said, very early in development. But the skin cells, as well as some nerve cells and some pigmented cells, are produced from ectoderm embryonic cells. Mesoderm cells produce different types of muscles, including cardiac muscle and skeletal muscles, as well as the blood cells, and tubule cells of the kidney, as well as some pancreatic cells. And the endoderm primarily forms other types of pancreatic cells, thyroid cells, and lung tissues. And then you have the germ cells at the end, forming either sperm or egg cells. So when the cells become cancerous, they then take on those gastrula-like or embryonic type characteristics, including rapid cell growth, influencing cells around them, growing blood vessels towards them, and also metastasizing. Although some cell types metastasize more readily than other cell types. Many types of blood cells metastasize readily, so you get lymphomas as well as leukemias. So starting from a multipotent hemopoietic cell, stem cell. Then you generate these common myeloid progenitors and common lymphoid progenitors, and so those become either the leukemias or the lymphomas, respectively. So they often will then take on those early type characteristics of the cell that was one of either the stem cells or a progenitor cell. And that includes rapid proliferation and growth. And so many times in the case of some leukemias, the first thing that's noticed is there's a high white cell, and often high concentration of white cells in the blood. And there's also often sweating and fever because some of the basophils, neutrophils, eosinophils, and mast cells produce those characteristics, including fever and inflammation. Whereas the lymphoid cells produce other types of characteristics, including a high white count as well. This graph shows the cancer incidence in the U.S. Based on a rate per 100,000 people. So you can see the highest incidence is for breast cancer, and then next for lung cancer, although that's been coming down as people quit smoking. Colorectal, melanoma, nervous system, uterine, cervical, ovarian, and then a lymphoma, some pancreatic cancer, etc. So this is just the incidence, the number of people being diagnosed with those diseases. These graphs show the mortality or death rates for those cancers in males on the top and females on the bottom. So you can see, even though breast cancer is the most prevalent cancer, it's not the most, it doesn't cause the most deaths. So in males, lung cancer causes the most deaths, and most of those are related to either smoking or working in environments such as coal mining, asbestos mining, where you have a lot of dust in the air, carcinogenic dust. For males, then prostate, colorectal, pancreatic, hepatic, and then a leukemia. For females, lung cancer is still fairly high. Again, most of that is related to smoking and other environmental exposures. And then secondly is breast, and then colorectal, pancreatic, ovarian, etc. So this is a little flowchart of who gets breast cancer, what proportion of the population gets breast cancer. And this is just for women, although I want to mention that men can also get breast cancer, and it turns out to be usually more deadly because there's less surveillance of finding it early. So in women, if you look at all women, 88% of women will have no breast cancer at all. About 12% of women will have breast cancer sometime in their lifetime. Of those, about 3/4 are sporadic cancers. That is, they're not related to any familial connection at all, but they're just from random mutations in the breast cells, breast tissue cells. About 25% of those are familial cancers. The largest proportion of those are mutations in multiple low prevalence genes, that is, or low penetrance genes. That means that there's a predisposition to developing cancer, but it's by no means assured. And so most of those women won't get cancer, but a proportion will. Then there's 5 to 7% that are high penetrance. That is, the cancer risk is greatly increased in these women, so that their chances of developing breast cancer are high, and their chances often of getting uterine not uterine, but ovarian cancer are also quite high. So both of these, the familial cancers, can be affected by lifestyle and environmental risks, so that they can certain measures can be taken to mitigate against getting or developing a cancer. One of these, the 5 to 7%, the high penetrance, that was one thing that Angelina Jolie found out that she had when she discovered she had breast cancer. Her mother died of cancer, having breast and ovarian cancer. So she chose to have a mastectomy so that she would not have to, or would not have the fear of developing a very possible breast cancer. Most cancer cells have several characteristics. If you look at a normal cell, usually a normal cell will stretch out, and it will eventually be inhibited by cells around it. So it'll take on a specific form for a specific cell type. When cells become cancerous, often they will round up. That is, they'll lose that characteristic shape that they should have if they were a normal cell. So they'll round up. The same thing happens when cells are dividing, by the way. So if a cell is dividing, they often round up, divide, and then stretch out again onto the subsurface, for animal cells anyway. If you look at a group of normal cells, they will stretch out, then they'll be contact inhibited. That is, if they encounter another cell, there is cell-to-cell communication, and they will be inhibited by those interactions to grow any further, so that in animal cells, they'll form a monolayer in tissue culture. So you'll have only one monolayer, and the cells will be fairly well stretched out one from another. Cancer cells, on the other hand, are not contact inhibited. If you look at the nuclei also are not the same size. Sometimes you get micronuclei, little pieces of the chromosomes broken off and forming separate nuclei. And furthermore, they're not contact inhibited, so they keep growing until they've reached the limit of the Petri plate, and they'll start to pile on top of one another and form not just a monolayer, but a multi-layer. And if you let them keep growing, they can actually grow out of the plate, if you give them enough nutrients anyway. So here are a couple of micrographs of normal fibroblasts in culture on the top, so that you can see they're spread out. They stop growing when they contact one another, and that's their standard way of interaction. You can also see a few cases where they've rounded up and they're ready for dividing, or they have divided already. On the bottom are cancer cells, and you can see they're not contact inhibited. They start growing together, and they grow more and more. And you can see the cells are not the same size. And if you look at the nuclei, they also vary in size. And if you could stain the nuclei for DNA, then you'd see some also contain micronuclei, which is very characteristic of cancer cells. The development of cancers takes several steps. So the initiation of a tumor starts with a single mutation, and then that can progress to more and more mutations, eventually forming a group of cells that are then classified as being cancerous. That is, they'll start to invade other tissues. So if you start on the left over here, you get a single mutation. The cells still look normal. There's no real danger in those cells. They continue to divide, and if you get a second mutation, now the cells in the center have both of those mutations, but you still have a ring around that containing cells with the original mutation. Additional mutations are normally caused by at least one or several key genes affecting the either the correction mechanisms for the mutations or directing mutations themselves. So now you have 3 mutations in the center, cells with 3 mutations, a ring around that with 2 mutations, a ring around that with 1 mutation. And then this continues until you get more and more mutant cells and more mutations in that central group. This is one reason why when they do surgery on tumors, they don't just take out the tumor itself, but they also cut tissue out from the surrounding areas, because those often, almost always, contain some of those original mutations that caused the cancer in the first place. And so if they cut too close to the tumor, often at a later time they'll see new tumors arising in those margin areas where the original tumor was dissected. So as you get the tumor progressing, it'll first be benign as it grows and becomes more or less a lump. But it hasn't invaded other tissues. Once it starts invading other tissues, then it's known as a carcinoma in situ, in that it has started to invade other tissues and become more dangerous. Cancer cells often, or tumors themselves, often will attract blood vessels to themselves so they can get a blood supply, but also pieces of the cancer cells or tissues will break off and enter the bloodstream. Some of those can then move and be lodged in other tissues and start a tumor there. So you can actually have a colon cancer winding up in the liver. So you'll have a tumor in the liver that really originated from colon cells. Once the cells have metastasized, the tumor has metastasized, then control of the cancer is much more difficult, because now you have many different loci, and just surgery is not as possible in that case, because it's starting to move into different areas, and then more chemotherapy and radiation methods are used. So it becomes much more dangerous once the tumor has metastasized, the cancer has metastasized. This shows the progression of colon cancer and staging of those colon cancers. So almost always after they're diagnosed, then they're staged. So stage 0 is carcinoma in situ. That is, there are abnormal cells now, but they haven't invaded any other tissues. Stage 1 is where they start to invade other tissues. Stage 2, they invade further into different layers of, in this case, the colon. Stage 3 is when they've gone all the way through most or all of the layers of the colon, and then some of the cells can be found also in the lymphatic system. And finally, stage 4 is where it's not only found in the lymph nodes and lymphatic system, but then some of the tumor has metastasized, moved to distant organs. Most of the time, the first organ for a colon cancer to go to is the liver, and in many cases, it's the liver. All cancers involve mutations of specific genes, many different categories of genes. So in the case of common changes in carcinogenesis, for instance, in a colon cell. So you have a normal cell and you get a mutation in what's called the APC gene, which is a gene for cell cycle control. This causes hyperproliferation of those cells, and then you get an early adenoma. And then the KRAS mutation, which is a GTPase signal transduction gene, can mutate, often mutates, and you get an intermediate adenoma, and you can see the adenoma up above. It's a little bulging out of that tissue. Then a DCC mutation, which is a transmembrane receptor protein, late adenoma, and then p53, which controls apoptosis. So now the cells are more or less immortal. Carcinoma is formed, and then others, and then you get metastasis. So it's multiple mutations in all cancers, actually. So here's one of the examples of a pancreatic cancer, and they don't all have the same progression of these mutations, but this is just in one case. You have first a mutation in mucin 1, and then the next mutation is telomere shortening and a KRAS mutation, followed by more mutations in PSCA and another mucin gene in fashion, which sort of adheres cells to one another. Then one to a P16, which is a common mutation in cancers, mucin 1 mutation here, cyclin D1, which controls replication in the cell cycle, P53, which controls apoptosis, BRCA2, which senses changes in DNA mutation, TOPO2, and some other genes, and then eventually a mutation in mesothelin, which then allows those cells to invade and eventually metastasize. This diagram summarizes some of the mutations in colorectal, prostate, and pancreatic cancers, going from normal cells through dysplasia, an adenoma or neoplasm, carcinoma in situ, invasion, and then eventually metastasis. So at the top you can see LOH and gains, so that's in the karyotype. So gains means that the chromosome, that particular chromosome, has gained sequences, gained DNA fragments and sequences. LOH means loss of heterozygosity, meaning one of the chromosomes has lost pieces off of it, and so you've lost some of the alleles for those particular genes. And then you can have methylation pattern interruptions and then gene mutations. So in the case of colorectal cancers, the APC mutation is frequently encountered in those, as well as P16 and DCC, and you can see below that those are tumor suppressor genes. KRAS is also common in colorectal cancer as well as pancreatic cancer, and that's a signal transduction pathway gene. Also, all of these have a P53 mutation, so it's a loss of function of P53, and that essentially makes all of these cells immortal, and then they just keep growing and multiplying. The genes in dark green are growth regulators, mutations in growth regulators. The orange are genes important in DNA repair, such as BRCA2. And then you have other categories of genes down below for transcription regulation, cell proliferation, control of apoptosis, detoxification, etc. But you can see from all of these that there's sequential mutations in every one of them, so that you start with one mutation, two mutations, and then more and more and more mutations, making that tumor act more irregularly and eventually becoming much more dangerous, much more possibility of lethality. The types of genes that are mutated in cancer cells are of a few types, including transcription regulation, cell adhesion and motility, signal transduction, so those make up about 60% of the cells, and then transport, intracellular trafficking, RNA metabolism and cell metabolism, and some other functions make up the other about 30% or 40% of the genes that are known to mutate to cause cancers. And if you look at different types of cancers, as far as the types of genes that mutate, the proportions are very similar. So cell adhesion and motility, signal transduction, and transcription regulation, again, make up the largest proportion in both breast cancers and colorectal cancers, as well as other types of cancers. And then you have transport, metabolism, intracellular trafficking, RNA metabolism, and others, making up, again, about 30 to 35% of the genes that mutate in cancers. This is a diagram showing some of the major molecules involved in carcinogenesis. So you can have growth factors such as these up here, and the growth factor receptors down here, and then signal transduction is a major cause of some of the loss of contact inhibition in cancer cells. So if you can't get the signal recognized, then the cell sort of goes out of control. Then you have regulators of growth, adhesion molecules such as the cadherins, which are cell-to-cell recognition, APC, which is a tumor suppressor, and then some of the molecules that act inside the nucleus, including cell cycle regulators, DNA repair recognition proteins such as BRCA1 and BRCA2, cell cycle inhibitors, and transcription factors. And then apoptosis genes too, genes that control programmed cell death, including BCL2 and P53. This shows the pathway and activity for one protein, the RAS protein. So when a growth factor is attached to a growth factor receptor, then that signal changes that protein, and it activates a bridging protein, which activates RAS, and then RAS, which normally holds a GDP in a certain location, exchanges that for a GTP, and that activates RAS. RAS then phosphorylates other proteins, which activates them. And some of those activate the MAP kinase pathway, that is the mitogen activated protein kinase pathway. And many of those things that are activated become transcription factors that then turn on the cell cycle, replication, etc. If RAS is faulty in some way, it will continuously phosphorylate and activate the cell, so that in cancer cells often that happens where there's continuous activation, overactivation of that cell. Here's a more complete version of what's going on there. So there's the RAS in the center, which will activate RAF, which activates this MAP kinase pathway. So there's a receptor on the outside that once the growth regulator is attached to that, the protein then signals SHC, GRB2, and SOS, and that activates RAS to accept the GTP in replacement for the GDP, and that phosphorylates RAF. And it's also activated by a different pathway that includes SRC, SRC, which is one that was first identified in a sarcoma. And then all of these tend to activate transcription factors, which will then initiate activation of the cell to replicate and undergo division and become a cancer cell. Here is more detail into that process as well as others, and all of the proteins that are known to be mutated in certain cancer cells or many types of cancer cells. these are in red, and all the black ones are just other proteins involved in those pathways. So you can see there are growth factors on the outside. There are cell surface proteins, receptors, cadherins, integrins, RTK. And then there's RAS in the middle there that also is affected by ABL, and there's an Abelson cancer that's known. There's SARC over there, which was first identified in a sarcoma. And then those all feed into this Jun, FOS, and MYC transcription factors. And also you have on the other side proteins that affect the cell cycle, cell proliferation, cell damage, cell death, abnormal sensors of DNA damage, such as BRCA1 and BRCA2. So a whole set of signal transduction pathways and others. This just simplifies things a little bit so that you have on the top the regulators, the cytokines, growth factors, things like that, hormones, which interact with cell surface proteins. And so those are the receptors there. And then you have these signal transduction pathways where that signal then causes changes inside the cell, which are normally pretty complex pathways leading to a function of that cell of one form or the other, including changes in gene expression control through activation of transcription factors such as MYC, FOS, and Jun. Cell cycle control regulators such as cyclins, and also cell damage regulators, including BRCA1 and BRCA2. And then those often will signal, if there's too much damage, they'll signal apoptosis genes and proteins, including P53, BAX, and some others. And you'll get cell death if there's too much damage. Now, if any of those are mutated in a cell, then it can cause the cell to develop into a tumor and a cancer. Again, this is a more detailed view of that, showing some of the growth factors on the outside, cell surface proteins in the membrane, and then things that react with because of those interactions, including RAS, which is there. And then a signal transduction here, and eventually leading to changes in transcription, differentiation, and apoptosis. This diagram shows that when there is DNA damage, how especially different systems such as ATR, but also BRCA1, interacts in several different places in this pathway, and if there is sufficient damage, it will then signal P53 to affect the cell cycle by affecting CDC proteins, and will cause the cell to undergo programmed cell death. So any of the proteins in this whole network, if they mutate, can cause the cell not to repair itself and not to die and start dividing to form a tumor. So as I indicated, not only in this lecture but in others, DNA repair is very important, and both BRCA1 and BRCA2 are involved in signaling DNA repair, and then if DNA repair is impossible or impractical, it will signal P53 to kill the cell. But in this case, you have DNA damage sensors. They will signal BRCA1, and it will then stimulate homologous recombination. It can stimulate other repair pathways, and again, if it's unable to repair or senses much more repair than can be accomplished, it will then signal P53, which will start the apoptosis process by stopping the cell cycle, and eventually the cell will die. So BRCA1 and BRCA2, especially BRCA1, they both are very important in recognizing DNA damage and then signaling the repair systems to repair the damage. So if they mutate, this can cause a cell that has a lot of DNA damage to go unchecked and keep replicating, causing more and more mutations, incidentally, and many times forming tumor cells, a tumor and breast cancer, as well as other types of cancers. Also, if P53 is not active, if it's mutated, then the BRCA1 cannot signal the P53 to start apoptosis, and that can also cause a cancer. This shows the some of the mechanisms involved in a specific type of cancer called APC, or adenomatous polyposis coli. So in the fetus on the left-hand side, the normal condition here is where you have activation by MYC, FOS, and JUN, which are transcription regulators, NB-catenin, all of which are leading to increases in cell division and growth. In the adult, however, you don't want the cells to divide so often, and you don't want as much growth. And in this case, the APC is activated, and the growth regulators are not activating MYC, JUN, and FOS. But APC is activating other proteins, which then phosphorylate the beta-catenin. The beta-catenin is actually broken down by a proteasome, so it also doesn't activate the gene expression. However, in colon cancer, the APC gene is mutated, and so even though MYC, JUN, and FOS aren't turned on, the growth regulators, by the growth regulators, the proteasome is non-functional because APC isn't activating degradation of beta-catenin. So the beta-catenin still causes increases in DNA replication and cell division and growth, causing the tumor cells. And then ultimately, as I've shown before, other genes are mutated, and the tumor just becomes more and more aberrant and has unregulated growth and immortality, leading to a cancerous tumor. Okay, that's the end of part 1, where I've talked about the characteristics of cancers, some of the progression of mutations to cause a cancer to develop, categories of mutant genes that lead to cancer, as well as some specific genes, proteins, pathways, and mechanisms that lead to cancers. Next time, I'll talk, in part 2, I'll talk about molecular mechanisms in cancer. So thank you very much.